NASA has revealed the first use of a tricorder to identify microbes in space.The genes and space.3 experiment aboard the International Space Station completed the first ever sample-to-sequence process entirely aboard the space station. It could one day be used to prove samples are of alien origin without having to send them back to Earth for identification first. The ability to identify microbes in space could aid in the ability to diagnose and treat astronaut ailments in real time, as well as assisting in the identification of DNA-based life on other planets, NASA says. It could also benefit other experiments aboard the orbiting laboratory.Identifying microbes involves isolating the DNA of samples and then amplifying, or making many copies, of that DNA that can then be sequenced or identified. NASA astronaut Peggy Whitson conducted the experiment aboard the orbiting laboratory, with NASA microbiologist and the project's principal investigator Sarah Wallace and her team watching and guiding her from Houston. The investigation was broken into two parts: the collection of the microbial samples and amplification by polymerase chain reaction, PCR, then sequencing and identification of the microbes. As part of regular microbial monitoring, petri plates were touched to various surfaces of the space station. Living bacteria have been found on the outside of the International Space Station, and they may be extraterrestrial, according to one cosmonaut. Russian engineer Anton Shkaplerov said the microorganisms were not there at the launch of the IS in 1998, and so likely flew from somewhere in space. The bacteria are now being taken back to Earth for further study after initial tests aboard the orbiting station show they are harmless to humans. They took samples from places where waste fuel accumulated and from obscure parts of the station. Previous experiments have shown bacteria can shapeshift in space. Pictured E. coli. Sample cultured on Earth, left, and E. coli. Sample cultured in the International Space Station, right. Rather than being killed off by the drugs, the bacteria in space responded with a 73% reduction in cell volume size, while growing the cell envelope to protect it, and now it turns out that somehow these swabs reveal bacteria that were absent during the launch of the IS module, said Shkaplerov. That is, they have come from outer space and settled along the external surface. They are being studied so far, and it seems that they pose no danger, the Russian astronaut, who will take his third trip to the IS in December as part of the Expedition 54 crew, said. Working within the Microgravity Science Glovebox, MSG. About a week later, Whitson transferred cells from growing bacterial colonies on those plates into miniature test tubes, something that had never been done before in space. Once the cells were successfully collected, it was time to isolate the DNA and prepare it for sequencing, enabling the identification of the unknown organisms, another first for space microbiology. An historic weather event, though, threatened the ground team's ability to guide the progress of the experiment. We started hearing the reports of Hurricane Harvey the weekend between Peggy performing the first part of collecting the sample and gearing up for the actual sequencing, said Wallace. When JFC became inaccessible due to dangerous road conditions and rising flood waters, the team at Marshall Space Flight Center's Payload Operations Integration Center in Huntsville, Alabama, who serve as mission control for all station research, worked to connect Wallace to Whitson using Wallace's personal cell phone. With a hurricane wreaking havoc outside, Wallace and Whitson set out to make history. Wallace offered support to Whitson, a biochemist, as she used the Minion device to sequence the amplified DNA. The data were downlinked to the team in Houston for analysis and identification. Once we actually got the data on the ground, we were able to turn it around and start analyzing it, said Aaron Burton, NASA biochemist and the project's co-investigator. You get all these squiggle plots, and you have to turn that into As, GS, CS and TS. Those As, GS, CS and TS are adenine, guanine, cytosine, and thymine, the 4 bases that make up each strand of DNA and can tell you what organism the strand of DNA came from. Soon after, the samples returned to Earth, along with Whitson, aboard the Soyuz spacecraft. Biochemical and sequencing tests were completed in ground labs to confirm the findings from the space station. We did it. Everything worked perfectly, said Sarah Stahl, microbiologist. Developed in partnership by NASA's Johnson Space Center and Boeing, this national lab-sponsored investigation is managed by the Center for the Advancement of Science in Space. Genes in Space 1 marked the first time the PCR was used in space to amplify DNA with the minute-per thermal cycler, followed shortly after by biomolecule sequencer, which used the Minion device to sequence DNA. Genes in Space 3 married these two investigations to create a full microbial identification process in microgravity. It was a natural collaboration to put these two pieces of technology together because individually, they're both great, but together they enable extremely powerful molecular biology applications, said Wallace. NASA has begun a crucial experiment to see how bacteria such as E. coli could affect astronauts. The space agency shot a sample of the bug into space from the International Space Station. They believe that in zero gravity, the bug could have an increased resistance to antibiotics, which could cause major problems for astronauts. Blast off. After arriving at the International Space Station, crew worked with ground controllers to release the satellite into orbit, and it is programmed to automatically begin its experiment. The experiment: if we find resistance is higher in microgravity, we can do something, because we'll know the gene responsible for it, and be able to design countermeasures, said A. C. Matten, Principal Investigator for the Camps of Investigation at Stanford University in California. If we are serious about the exploration of space, we need to know how human vital systems are influenced by microgravity. Scientists believe that bacteria like E. coli may experience stress in microgravity. This stress triggers defense systems in the bacteria, making it harder for antibiotics to work against them. Bacteria on Earth do something similar by developing a natural resistance to traditional antibiotic treatments.